Okay, guys, let's have an overview of flow cytometry. Sorry. So this flow cytometry is something that we have always come across it, as I've been talking about various laboratory tests that could be done for various disease conditions, right? Especially when we are talking about blood physiology and all of that. So this flow metry is how the test is. You just have to do it. Yes, two things mixed, then they put in that container. So as they come out, they pass through a beam of light. So the ones that are positive will now go one side. The ones that are negative will now go one side. That's the summary for it. Okay. You see how it is. Okay. So let's move, guys. You see that flowmetry is a fascinating and powerful technology which is used in the fields of biology and medicine. All right. So what is the flowmetry? We said that it's a technique used to detect and measure physical and chemical characteristics of a population of a particular cell or particular particles. Alright, so this is accomplished by what? Suspending cells in a stream of fluid, then passing them through what? An electronic dictation apparatus like this. Alright, you put the sample through here. Alright, now this is a laser beam and they pass through it. Do you understand? So how does it work? First of all, there's sample preparation so that cells are often tagged with fluorescent markers that bind to specific cellular components. And this will now allow for what? Targeted analysis. Fluidic system, which that this transport the cells in a single file through a laser beam, ensuring that data is collected from individual cells. As individual cells are passing through that laser beam, data is being collected. Laser interrogation, we say that the laser excites the fluorescent markers, causing them to emit light at different wavelengths. Dictators, this capture and measure the emitted light at various wavelengths. Computer analysis, the data is analyzed to quantify and characterize the cells and particles in the sample. Alright. So application is applied in medical diagnostics, so it's used for counting and sorting cells, diagnosis of healthcare disorders like Fanconi anemia, monitoring the effectiveness of treatments, particularly in what? Immunology and hematology. Immunology is basically the study of your immune system, trying to see if your immune system is still competent and all of that. Okay, then I think basically that's it. Also for research, to analyze the physical and chemical characteristics of cells, study cell cycle distribution, and measure levels of cellular markers. Then cancer dictation, it identifies cancerous cells, then assess the proportion of different types of cells in a sample. Immunophenotyping, it assesses the type of cells present in a sample and their state of activation. Alright, so this is it for flow cytometry. Bye for now.